Don't see you.
Hello, everyone who's joining us today for our first in our webinar series on composting in the time of COVID-19. We're very excited to be presenting this today. My name is Kerry Oceans. We're just going to wait a few minutes while folks log in. We've had an unbelievable response to this webinar. Well over 800 people have registered. And so we'll expecting a really nice turnout, and you're in for a treat on today's webinar. We're still just a couple of minutes early, so we're not going to get started here. We'll give people chances to get logged in. We have people coming in from all over the world for this webinar, pretty much almost every state, and last I checked, 26 different countries. So again, we'll give people a few minutes to get logged in, and then we'll be getting started. Again, my name is Kerry Oceans, and I'm here to welcome you all to our first in our webinar series about composting in the time of COVID-19. Today we'll be looking at reviewing safe procedures for composting of feedstocks with possible contamination. Now I'm on the right slide. Okay, we've got almost 400, over 444, 445 people. So we're going to go ahead and get started with this webinar. Before we get started, just a little word from our sponsor here. For those of you who are not familiar with the U.S. Composting Council, we think of ourselves as the voice of the U.S. composting industry. We're here to promote composting at all its levels and support our members in helping us all to have a cleaner environment. A few housekeeping items to attend to before we get started with the first presentation. Everyone is on mute. That's to help keep the airwaves clear and keep the audio nice and clean. Some of you have already found a question and answer box. We will be, I'll be fielding those questions. We'll probably hold most of the questions to the end, but if there are specific questions for a speaker that we can sneak in between the two, we'll certainly do that. Many of you have asked about if it's if the webinar is being recorded, and yes, it will be recorded, and it will be available for on-demand viewing and that'll be at our website. And and on our website, if you go there at compostingcouncil.org, you'll see the menu of items across the top. If you click on the research educate one, the very top one will be the COVID-19 resources. So that's where you'll find these and lots of other great resources that we are collecting for everyone to have available to help answer questions during this time. And so just before we get started with today's webinar, just a little word about what else is coming up in our series. Of course, this is all dynamic. None of us had this planned 2 months ago or even a month ago. And just to do a quick shout out to not only our team at the Compost Council, but the folks at the Institute for Local Self-Reliance and the Composting Association of Vermont for helping us pull these together. So a week from yesterday, next Tuesday, we'll be talking about composting as an essential service and have a panel of operators from around the country, some smaller ones, some larger ones, some from the East Coast, West Coast, and Heartland. So really nice variety of folks that have agreed to step up and talk about how what they're doing, what kind of practices they're implementing to keep them and their customers safe and keep sales moving. On the following week, we'll be focusing on collection, and a lot of the questions that came in asked about collecting, especially for community composters. So we are having a dedicated webinar on that on the April 14th about for small scale food scrap collectors, whether it's by drop off at community gardens or community sites or collecting curbside. And then the last one we have scheduled now, although we may be adding more, is on how to sell compost to customers who are sheltering in place. So we've probably seen a lot of talk about the reemergence of the Victory Garden while folks are sheltering at home. And so certainly compost is a key component of any successful garden. So we'll be having some information on that as well. Okay, getting started with today's webinar. We've got two great speakers today. I'm going to first introduce Pat Milner with the U.S. Department of Agriculture. She's going to be speaking first and really focusing on the virus itself and what we know about the virus. After Pat goes, Jean Bonnetall from Cornell University will be talking about some of the practicalities around the site management and pathogen control, not just of the COVID-19 virus, but other kinds of pathogens you need to be aware of at a compost site and what you need to do to protect yourself. And Pat, I'm going to stop my share so you can load yours up while I introduce you.
Where did it go?
Okay, Pat Milner is has been working at the U.S. Department of Agriculture, the Agricultural Research Service since 1976, and she was part of the team that helped develop the aerated static pile method of composting. She has expertise in microbiology of composting, compost products, food safety, and pathogen reduction, and she's been a really great friend to the industry for her career, and we really appreciate her being with us today. So go ahead, Pat.
Okay, so I'm going to talk about the virus itself, inactivation, and how that relates to the composting process. And so we'll get started here. This is a rendition of all the structures of the virus. Actually, to make a distinction here, the disease is named now COVID-19, but the actual technical name of the virus is SARS-CoV-2. And this derives from a convention for naming these viruses that is an international convention. What's important about this is that these red colored structures are part of how the virus actually uses its own structure to attack and attach to gain entry to the human respiratory tract. These red structures are called spikes. They're glycoproteins, And those are actually considered part of the virulence factors. And those spikes actually attach to receptors on the host cells. And this is the initial part of the process that allows the virion to gain entry into the respiratory tract cells. Once it's in the cell, it begins its replication process. And we'll show you that in a little bit. This spike protein, it has been sequenced and has been documented to be 98% identical to the spike protein of the bat coronavirus, and which is why you've probably heard over the popular press that this may have originated from bats. And there's a whole history of other coronaviruses that are also have this same sort of pattern on these spike proteins. For each of the slides, I'm going to present where the information that documents what I'm talking about came from. And so that'll be in these references that you see cited in case you want to go to those references and have to make a documentation of that in the future. So just to kind of encapsulate this cartoon of the virus with these spikes here, each of the spikes themselves has a couple of parts to it. This is the part which actually hooks on to the place on the host cell, but there's this other piece of the stem stalk, which is a different part of the protein. And so it's a stepwise process to actually gain entry into the host cell. So this gives you somewhat of the cycle. So once it attaches to the host cell, there's a number of different steps that it has to go through. And I'm not going to go through all of these and what they do, but just look at the idea is that there's a complicated series before it completes its replication and then is excreted and goes off to create further havoc. And this is elucidated in this reference here. And this diagram was presented in this reference. So you can follow that in detail, and there's a lot of explanation there for those who are interested. In terms of the inactivation of the coronavirus that induces basically all the types of SARS coronavirus respiratory syndromes that have been seen over a number of years. And some of these were the original SARS virus outbreak that occurred, and then some subsequent ones which occurred in the Middle Eastern areas, but they're all very closely related. So at the time that the original SARS happened, they actually, Darnell and that group, they looked at what was the tissue culture infectious dose. So one needs to know something about, well, how many particles and what kind of time and temperature will kill this thing. And so this, they looked at 56 degrees, 65 degrees centigrade, and all three of them, and up to 75 degrees centigrade. And so what they're seeing is that the number of logs per milliliter that you can actually inactivate these, it drops off very quickly once you're up at temperatures that are akin to what we see at these temperatures. So we can see that for like cooking type temperatures, which some of these are in terms of, if you translate this to Fahrenheit, you'll see that these can be inactivated in a very, very short period of time. I'm sorry, the USDA sends us these little messages at periodic intervals, and let's see if I can get rid of it. So looking at a variety of these coronaviruses that have been isolated over time, the MERS virus and several of the different SARS viruses and these other intermediate ones that were associated with different outbreaks in the past, different people looked at these log values. In other words, how much, how many logs would be decreased, how many logs of destruction would be decreased at different temperatures. And so they looked at 30 degrees centigrade, you don't get very much destruction there. But as you increase and you get up to 60 degrees centigrade, and this looks like almost 65 degrees centigrade, then you can get quite a number of log reductions. So that's where we come to some understanding of, well, once we see the whole linear fit models for all these different data points, we see that the best destruction occurs with these very higher temperatures in very short periods of time. So some of these studies also looked at whether if there were additional things that might be protective, such as proteins. So if you have at 4 degrees centigrade and you have no protective protein, in this case, you get you get no change, essentially. You get very little kill at the after 30 minutes of exposure. And that's the same if you have the protein added. It's not affecting. But if you increase the temperature, so with no protein added, you have lots of reduction. But when you have the protein added in, it protects. And so you don't get very much reduction in, even if you're at the higher temperature. And at 60 degrees, you get a lot of reduction regardless of this protective additive. So what this continues to look like is that the higher the temperature that you can expose the virion to, then you can get a greater amount of reduction. And so those are the kinds of temperatures that you'd want to maintain in the compost, because in most composts, you're going to have something that's in there because you've got other microorganisms and other substances in there that could act as protective. So you want to operate at the highest temperatures possible. So in summary, some of the more recent things that have come out, and this was very recent, and there's been a flush of new things that have come out. The current virus survivals that have been evaluated environmentally for how infectious they are is up to 3 hours in an aerosol, up to 4 hours if it is on a copper surface, up to 24 hours on a cardboard surface, and up to 72 hours on plastic and stainless steel. So what, well, meant to go backward. Yeah, not good here. What does this mean in terms of composting facilities? Well, if you're indoors, someplace like in the first hall where you have the material is delivered, if it's in a covered and area there could be some aerosols. So the raw materials, when they're brought in, if they would contain anything that might generate some kind of an aerosol, that would be an area that you would want to have some PPE for operators and personnel that are around there. It also might be an area where, because of the metal on the equipment that your operators are dealing with, maybe they're like, for instance, the steering wheels or whatever handles are on things, might be a good idea to have some sanitation on those touch points, because the aerosol could land on that. And so there's another thought to keep in mind about that. in terms of food, people have been really concerned about whether there might be an issue with food. And basically that goes back to the sanitary operations that are normal for food handlers. So the main way that food might become contaminated is if there's some droplets that from a person who has contracted the illness, and they are sneezing or coughing or handling the food when they haven't washed their hands thoroughly enough yet. So it really is a respiratory tract illness, but because of people's hands being involved, that really brings it to this way of needing to be very diligent about the hand washing thing. So there's no real evidence that the food is in itself a transit means for spreading this, unless, of course, the food gets contaminated by somebody who has not used good sanitary practices. Of course, cooking, if you're cooking the food up to like 63 degrees for at least 4 minutes, this will inactivate it. That remains to be that's the internal temperature as well. So if you're making hamburger patties or anything else, it's just like any other food handling situation. You need to observe very sanitary practices for food handling. Just put this up to remind people about all of these sanitary practices. And this is, probably you've seen this in a number of different formats, but this is really pretty standard now for people to understand and go through. So keep washing your hands. In terms of how all of this relates to the operations at a composting facilities, basically for 4 points, which USCC has already developed into their guidance. Workers should wear appropriate PPE when handling and mixing those raw feedstocks, particularly if it's in an enclosed or semi-enclosed building where the material is dropped off. Any equipment that is used for feedstock handling and mixing should not be used for the product after it's been through the composting, the peak heat, and curing phases of composting. And for screening or loading or finishing or any finished type product, you need to use clean equipment for that, no cross-contamination. The feedstock that is being received and mixed should be located in a downstream or physically separated area from that finished product material. And then the other piece of it is that how to handle the equipment. Think about how to clean up those buckets, the tires, how to keep from tracking material from one area to the other. If possible, steam cleaning of the equipment or other appropriate disinfectant. EPA has put out a number of lists for appropriate disinfectants that are suitable for this. Reminder, though, is that in order for a disinfectant to work effectively, the object that you're trying to disinfect has to be cleaned first, and then the disinfectant is applied. So if there's still dirt or mud or some other kind of material on the tires, you need to clean that off first, then apply the disinfectant. And here's the website for the USCC site. I think that's the end of it. If anybody has any other questions, then that's...
Yeah. So great. Thank you, Pat, so much. Yes, we do have a question I wanted to post to you now. This is from Brian Fleury. He said, would an aerosol be the steam or, okay, would aerosols be emitted from compost piles that are steaming or off-gassing. Is that an example of an aerosol?
No, that's steam that's generated from the condensate. You're talking, well, in this instance, we're talking about some kind of a droplet that would have already had the virion in it and then is forcibly ejected, like from a cough or from uh, some other forcible means like that. I'll paint you a scenario here. Is, let's say somebody coughed on some kind of food that they were preparing. So then they decided to throw that food away. Okay. They put it in some kind of a recycling container that they had in their kitchen. And then they put that out in some other collection system, whether it's their own backyard system or a community system or whatever, is that has some droplets on it. Okay. So that material has already been aerosolized, but it's now stuck to whatever it was on. Now, if that material is subsequently, I don't know, thrown up into the air by somebody's bucket loader or something, once these virions are on something, and there is a study out there that was done on one of these progenitor coronaviruses. People have done the study on lettuce, for example, and lettuce leaves. Once the virion attaches to something like that, it's very difficult to wash it off. So it's very difficult then, if you can't wash it off very easily, you can't actually just fluff it up from something very easily. It's going to take some special kind of force to dislodge it from there. And at the same time, when in a compost mixture, you do have lots of other organisms there and lots of other enzymes. And because those spikes, which are a key part of the whole process for initiating an infection, what you need is to think about is the fact that you've got proteinases around that are probably capable of kind of disinfecting or causing a structural change in that spike protein. So that spike protein is a really integral part of that whole process to get it into somebody. But the droplet is the key piece, and the steam is not full of those droplets because there's no force ejecting that steam from any surfaces.
Great. Thank you so much, Pat. Okay, we're going to move on to our next speaker now, and Pat will be around to answer more questions at the end. So our next speaker is Jean Bonital. Jean's with the Cornell University's Waste Management Institute. She's worked there on, well, it says here more than 20 years. I know personally it's been longer than that because I started with her back at Cornell longer ago than we care to think about. Currently, her time includes work on food scraps, manures, and carcass and butcher waste compost, education and research. She's also worked with the U.S. Forest Service, the National Park Service, EPA, the New York State Department of Environmental Conservation, and the landscape and greenhouse industries. And just to let people know, and as part of Jean's talk, there will be a link or two, and we'll be posting the slides as well as the actual video of the webinar. We'll also post the slides from each of the presenters in PDF form. So you'll be able to grab those links out of there. And tomorrow you'll get an email. Everyone who's on the webinar will get an email. Thank you for attending, but it'll include the links to those resources. All right, Jean, you ready?
Thanks. Carrie, that just means we're old. We've been here a long time.
I know.
So pathogens can cause disease. They include bacteria, viruses, nematodes, fungus, spirochetes, amoeba, and other parasitic organisms. Pathogens exist everywhere things are living, pretty much, even in hospitals, even in other places that we hope that there aren't a lot of pathogens. They need a pathway into the plant to cause, plant or animal, to cause infection. And they're looking for weakness, basically. The amount of exposure to infection combined with the susceptibility of the individual, so a plant or animal is weak, can result in illness. I work in some disaster response in terms of animal agriculture. And there was a highly pathogenic avian influenza outbreak in 2015 that affected 50 million turkeys and chickens west of the Mississippi. That we learned a lot from working with that virus and trying to compost and composting those animals through that compost. So I'll talk a little bit more about that later. There's an SOP, standard operating procedure at this link that Carrie will add to the chat box. I worked on a brucellosis suis outbreak, and that's the HPAI is zoonotic, the highly pathogenic avian influenza is zoonotic, and the brucellosis is zoonotic. One of the ways we found the brucellosis is a person got sick from the pigs. A person and a dog both got sick from the pigs. And then we realized that the pigs were infected and they had to be destroyed. Last summer, there was a fowl cholera outbreak in pheasants. This is a very limited population. These were wild pheasants that are in captivity, but those pheasants needed to be destroyed and then we composted those. And we responded, a bunch of us responded to the animal death, the animal destruction in Hurricane Florence in 2018.
Hey, Jean, before you move on, just a quick question came in. What is zoonotic?
Zoonotic means that we can get that disease. So we have to protect ourselves even more. When you have a zoonotic disease, it can jump that barrier. And really the highly pathogenic avian influenza, they did expect that one to jump to humans and become a pandemic. Luckily, that hasn't happened, and so we are experiencing a different one right now. In that, as I said, we had 50 million birds, turkeys and chickens west of the Mississippi that had to be composted or disposed of in a proper manner. Composting was chosen by most because of high water tables. There wasn't an easy way to bury that much flesh waste. So that had to be done. But we had to also look at all of the eggs, the eggshell, the feed that was served and not served, all the bedding, all of the litter, all of that material had to be sanitized, and that was all done through composting. USDA APHIS was the leadership on all of that and led us through that whole scenario, the whole episode, I should say. Let me just jump back once quick. So we were able to take care of that in this way. We also worked to look at pathogenic organisms in roadkill. So we did a roadkill research project. I'm not going to go through what it was exactly, but what we were trying to do was to see if DOTs were handling deer and composting deer instead of dragging them off to the side of the road or landfilling them or whatever. Would they be, what would we need to do to protect the workers from any deer issue, any pathogens from the deer? So we were looking for the most virulent pathogens in these. We enveloped these animals in wood chips, and I use this slide just to share with people. We generally look at all different types of feedstocks, clean and dirty. And if we look at these numbers here, the fecal coliform, the E. coli, the salmonella is pretty low, but all of the other, the fecal strep and the fecal enterococci in plain wood chips on the side of the road, the pathogens were very high in those. And this is just not to scare people, but to say we have to think about the feedstocks that we're using and make sure that we're taking proper precautions with our own health and animal health when we're using things like this. We looked at the hardiness of the different animals. The ones are the weakest. And if we put viruses in this hardiness rating, they're not even there. So they're a very weak organism. So they are much easier to kill. They're also doing a pretty good job of killing us right now. So we'd say, well, how can that be weak? But they are weaker. They respond to other sanitation. But Salmonella, E. coli, Campylobacter, Yersinia listeria, those are would be a level 1. Those are the easiest to kill. Then we go on to the second level, and Leptospira and Streptococcus and Enterococci are in that middle range. And then we go to Clostridium and Mycobacterium. And those are the hardier organisms. So we wanted to make sure that in the composting process, we were able to kill all of those and draw down the levels of all of those pathogens in the composting process. Mycobacterium has a waxy coating, so it tends to be a very difficult one to break down, to kill. And so we used that one, but we took that from cows and planted it in deer because it is one of those indicator organisms that we wanted to look at. The fecal coliform can grow and survive at 44.5 C. E. coli, it's in high numbers in birds and mammals. Salmonella, we actually didn't find in the deer. And what we were looking at in the deer was the gut of the deer. So we took the fecal sac and actually planted that back into the animals in specific ways. Fecal strep is in manure. Enterococci, it's antibiotic resistant. So these are tough ones. They're tough pathogens to look at. And we needed to look at those in terms of composting. And we actually were looking at those in terms of passively aerated compost piles. That's the method that we use for mortality, for all the mortality, partly because we don't want any aerosolization, especially with airborne disease like the high path avian influenza. We don't want any of that pathogen or the virus aerosolizing and moving. So we move things as little as possible. With the high path avian influenza, we composted those animals as much as possible in the barns. They never left the barns because we were trying to contain the diseases so it couldn't move out of that area. So we have a thing called PFRP, processing to further reduce pathogens, and most of you probably know this well. It looks at the time and temperature to reduce pathogens. And it's looking at those in aerated static piles and in turned windrows. As I said, we had to work out the protocol, and that SOP works that protocol out for a passively aerated windrow. We were leaving those windrows for a 2-week period of time before moving them at all. So we left the composting process to work for 2 weeks, 14 days, and then we went ahead and were able to move the piles, move the windrows out of the barn so that they could finish curing and they had a turn, so to speak. This is just a chart of the PFRP. The temperatures go up to a certain level. Actually, let me just jump to the, oh, there we go. To the 55C. And if we're looking at this white line here, this is the temperatures rising. They want to be above there, and they want to be above there, and they do go up and down a little bit and then come down. They usually don't come straight down like that as much, but we have the mesophilic period in here, which is the lower temperatures above about 90 degrees. Then we have the thermophilic, usually 104 to 158. And then things are, in this period here, things are curing and maturing. This is an actual temperature, and this is 3 probes, 4 probes, but 3 probes in some of those deer. And you can see these piles were started on November 31st. And you can see those temperatures just rising really quickly right up in the piles and above the 55C, so that the pathogen control is going to happen in those early periods up in this process here. We also will have, there also is pathogen destruction in other phases of that. So we were able to see that there was some good pathogen destruction below that line for a lot of organisms. Some of our piles up in Watertown, where it's extremely windy, did not get as high as we would have liked them to. They didn't meet the PFRP. And so it was good to see that we still, because we were testing, we were still killing those. The other probe is just ambient temperature. We looked at a lot of different things, but we looked at the fecal coliform and the E. coli in the deer feces, and those numbers went down very quickly. So we're seeing things drop down. This level, the blue line, those materials could be used in one's backyard, used for gardening and such. So we had good temperature, had good pathogen kill in there with that. We also used, when we were putting the SOP together, the standard operating procedure together for the avian influenza with APHIS, we were looking at the EPA 503 rules. They do pertain to sewage sludge, Class A and Class B, but we were looking at those because they give us some kind of baseline to go off of. And so we were really fortunate to have the 503s and the PFRP to use to go off into a field that we really haven't done that much work. we've been working, or people have been working in mortality composting for about 15 years. So, and this is the Class B is just, I'll let you read that slide, but the Class B was also used. So we have an idea of where we wanted to be with the standard operating procedure and when we could move things. PPE is interesting, and I use this slide here for PPE because this is the extreme. Probably this is totally protective. They'll put their hoods up on there, but everything's taped. They have Tyvek on or Tychem and masks and goggles and full head coverings, double foot coverings, really, because you have generally have a boot inside, a shoe inside of the boot. So you have a lot of protection, and usually lightweight clothes on because it can get pretty hot in those suits. I show this picture because it was on the same farm. The people that were going in to do some of the foaming and the euthanasia had full PPE on. The farmers were given some materials from the health department on the onset of the disease when we realized the disease was on that farm. And they had very little. So they had a dust mask, they had on like a lab coat. It's a Navy lab coat. It wasn't appropriate for them. They had been exposed. They were with the birds when the birds got it. So they had been exposed and everything, but I think we could have done a better job, and in subsequent situations, we did do a better job. This is one of the earlier outbreaks. But it really varies very much. Use of devices, I'll go into in a minute. But personal protection, appropriate personal protection for disease really depends on the disease. It's going to be different in every case. As I said, for the highly pathogenic avian influenza, we want to make sure that we have everything. We're really covering everything. And with the brucellosis sui, we had even more. We had face shields on top of that, and just a lot more protection. When we're in a regular composting situation, and right now we're not in a regular composting situation, but we're in a regular composting situation, we want to have our dust masks on. We want to have our eyes protected. We want to have our ears protected, hearing protected. Our faces should be protected in general. Our hands should be protected, but we're not trying to keep a disease out. In the COVID-19 situation where people are receiving materials, that's where there's potential. That in-between point is where there's potential to get contaminated. So it's really important in those in-between areas to have good masks, to have glasses, to have ear protection, to have, I would have head coverings, I would have my body covered. I mean, I would make sure that I had full coverage. I don't think we need to have Tyvek in these situations. Tyvek can be very hot. When we were in the brucellosis outbreak, we had to put ice packs inside of our, we put them in our pockets basically, so that you can keep cool enough to not pass out in a Tyvek suit at 90 degrees weather, which it was. So, you know, those are all things that we have to take into consideration. But if people are going to continue to receive, and I know some places have stopped, but if people are going to continue to receive materials that could be contaminated, I think it's important to take those precautions and make sure that you are well protected with PPE. I put in identify yourself on this, and that's kind of an interesting thing. But when everybody is dressed the same in Tyvek or something like that, and you don't know the people very well, you really need to write your name on the back of your Tyvek or whatever, so that people can yell if there's a danger or something like that. People can yell and say, "Joe, get out of the way," you know. So it's really important to do that in these outbreaks, these situations. Used PPE must be bagged, and it should be in an area. There's going to be, I should talk about that one first. Should be bagged as you're entering a clean area. So we have clean and dirty areas in our clean and dirty areas in an outbreak or in an area. And with our facilities, our everyday facilities where we're taking things in, it's going to be considered clean outside, and everything that you're taking in, putting on pad, all that kind of stuff should be considered somewhat dirty. There are all different feedstocks that we take, from manures to feces that we don't know we're getting, dog and cat feces, things like that, that are just thrown in with leaves, all different materials, and we just don't know what we're getting. And there is potential to have problems with those. But when we're in a serious area where we are in a disease outbreak, we need to bag all those materials. Those get thrown away in a specific place. Durable shoes and clothing are disinfected. Wash and change clothes prior to entering any living or food service areas outside the facility. So when people are going home to their kids, going out to the store, going out to whatever, just for a snack, it's pretty important to make sure that at this point that you're changing clothes, because we know that the COVID virus can persist on clothing. And by bringing that out, that can be a problem. Our dust masks, they're telling us, can be disinfected, can be sprayed with alcohol and stuff, and reused. And I think that's an important thing to do, is to make sure that if that's going to be reused, because we have a shortage right now, that it's disinfected. But don't bring it home. I think I heard on the news today that they're actually offering housing or hotels for people that are being affected and potentially infected, medical people, so that they don't have to go home to their families to take a rest. So that, you know, there's just a little bit more separation there. Wash your hands, your face, before eating, drinking, smoking. I tend to, one thing that I tend to do, which I know is kind of a strange thing, is I'll just take my lips, because people tend to lick their lips, especially after they're in a situation or they're drinking something. I usually take some hand sanitizer and go right across my lips as well when I'm in a situation where I think I could be contaminated. So, you know, just doing that. Wash hands before and after toilet use. Require that people use good dust masks and respirators if they're needed. Require, you know, areas can be dusty where things aren't processed. We have dust. I would consider any place where we have a lot of dust to be an area where somebody should have a mask on. And I can think of a lot of situations that you wouldn't, where it's just too dusty, and a lot of things can be breathed in and we have problems. Farmers can have brown lung just from breathing in some of the dust and some of the hay dust on a daily basis. And they end up with something that's called like brown lung, where that particulate matter starts getting in their lungs and filling their lungs. And that can happen in a composting situation. And not cause disease immediately, but cause disease in the long run. If a person has a compromised immune system, an employee or somebody that comes to visit a site, and at this point, people shouldn't be coming to visit sites if they're operating. It would be better if they were just, if the facility chooses to operate, use good PPE for all of their people, but forget about having visitors because it's just an unnecessary need. You know, they don't need to be in there at that time. If somebody has a compromised immune system, we are obligated actually to make sure that they are fitted with protective equipment. Or I would always suggest, and it's probably not a popular thing, that people that do have a compromised immune system not work in the composting, in composting where there's a lot of dust and potential bioaerosol and stuff like that. Phones and devices, I put that there because I was in a situation in one of these outbreaks where a piece of equipment broke down and we were in the middle of somebody dispatching or foaming animals, and the water supply died. And the person that was on the foamer just took out their phone and started playing games on it because there was nothing else to do at that time. And I thought, oh my. Most of the time, we don't even want to be using phones in these situations because they're near our mouths, they're near our faces, and just put them down. Forget about it. Phones and other devices that we might be using, we want to make sure that they're clean and they're not right near our faces. Managing feedstock and equipment to reduce contamination. One thing for health and safety is train people to know where all people are for personal health and safety. People get run over, people get left in places, people get stuck in places. We need to know, you know, just for that safety issue, we need to know where everybody is all the time. Use a clean bucket to move finished material. Some facilities do have multiple buckets and use those for different things. Not that long ago, we used to feed animals with the same manure bucket. So we haven't always thought about clean and dirty materials and how we can keep these things clean. Turn windrows the oldest to the youngest. So you don't want to contaminate those, and the youngest piles might have more pathogen in them. So we don't want to bring it that way. So you always have to think about it in a cyclical way. Sanitize all equipment used in the dirty or contaminated at the clean and dirty line. Sorry, I must have put that in twice. Control traffic patterns, use good signage. And avoid driving on piles. When we're driving around sites, we're moving pathogens. We'll go through a puddle, we'll pick up some pathogens, we'll move them into a pile. That's an important thing to do, is to think about that. Another place I'll just give you the quick example of is you can watch the milk trucks. If a disease is going from farm to farm, it may be on the milk truck tires, it may be on the picking up bird tires, or the veterinarian's tires. So that's why most facilities, most farms say no cars beyond this point. You know, some of the vehicles can be beyond that point, but a lot of things shouldn't be. And that's where I'll leave you today.
Okay, thank you, Jean. Let me make sure that we've got everyone. Let me unmute Pat, and then we've got some really good questions for both of you. Pat, can you hear me?
Yes.
Okay, good. I'd say.
Should I stop sharing at this point?
Yes, you can stop sharing. Thanks.
Okay.
Okay, the first question and the most frequent question that came up was how long COVID could last on a piece of food. So, you know, someone let's say an infected person coughs on their lettuce at home, and that piece of lettuce goes in the bucket, and a community composter comes to collect that. Is that virus still viable on that piece of lettuce for how long? And that's just an example, but in general, how long can the virus last on organic matter at ambient temperatures?
Well, first of all, nobody's done those kinds of tests in a controlled setting because they haven't done these isolated, they haven't isolated the organism in tissue cultures and done that kind of test. Everything is really focused at this point on the human diseases and so on. So the environmental part with regard to that food that's taking a secondary status at the moment because people are not thinking that it's transmitted on food.
Mm-hmm.
But insofar as where the food ends up, it makes some sense to me that if you're putting it in, let's say, a compost or some kind of a organic situation, where you collect all the material, there are a whole lot of exoenzymes that are there, and proteinases are chief among those. And although the material isn't going to heat up right away until it gets into a large enough mass, you can't count on the heating process. So the only other way that it's going to start to be destroyed is through some kind of enzymatic attack on those spikes. And that's going to depend on what the proteinases that are there, because that's the first area that's going to happen. And we don't have any evidence of, how long that's going to take. It's going to be very dependent on the situation that the lettuce or whatever the particular food is. There's always other microorganisms around, and other microorganisms produce these proteinases, but the efficacy there's no tests on that right now. So we don't have any data. So I think it's safe to assume that, under ambient temperatures, if you take something that is like the longest survival so far, we know that on, let's say, stainless steel, it'll survive for 72 hours on stainless steel. Well, it's probably not going to survive any longer than that, but if you count on that, then I think you've got some idea of how to gauge that. But it's going to depend on the situation.
Okay. So, but I think
it could be shorter. It could be shorter than that.
One thing that I learned with the avian influenza, the high-path avian influenza virus, is it could survive in puddles for days to weeks. And that made me think pretty hard about, okay, if it's the in-between place, it's not heated up, as Pat was saying, but it's in that in-between place, I have a feeling that we might find that it can stay on food that's been contaminated.
Okay, well, I guess it seems, I'm sorry, I have to interject because if the longest it lasts is on stainless steel is 72 hours, it seems like it can't last longer than that in those more active environments.
Yeah, as Pat was saying, it hasn't been checked.
Right.
So we don't know. We just don't.
We don't have there is no definitive answer. We're just basing this on what we know so far.
And Pat, what about, so people have asked about other kinds of disinfection like ultraviolet or pH changes. How would those affect virus survival?
Yeah, I think I've seen some pH range. It depends on how susceptible that protein spike is to a pH change. So you're talking about needing to go to a pH that's probably 10 on the high side or less than 4 on the low side you've got to get to some extremes of pH, which are not something that you're going to get unless you've got some strong acid or some really strong base, which, that's probably not going to happen. The basic core, that round part of the structure, that's a lipid layer. So that's why soap works so well, because it gets in when you're scrubbing your hands with soap, you're basically introducing something that's going to solubilize that lipid layer. So it's not attacking the protein part per se, but it's destroying that other part. So that's another piece of it, but that's not something that you're going to find in concentrations enough in a compost pile or a waste pile. You'd have to introduce that by some external means. As far as UV, I'm not sure whether we have any data on these for UV, but again, you've got to have, if you're depending on solar radiation, that's going to have to be a pretty exposed piece of material to be able to contact that. So I'm not sure how effective that would be in all cases. When you're talking about something like food pieces or other kinds of structures, the structure itself can have a shape that would impair every element of it being exposed to the same density of radiation and time. So that may or may not be effective to the degree that you're hoping for. And I don't have any data on that for this particular virus.
Okay, well, we'll watch for that.
Yeah.
I have several questions about aerosolizing. One about if there's a misting system in a receiving building, and could that kind of increase the survivability of an aerosol? And then someone else asked if you're grinding, like a tub grinder or a horizontal grinder, that if the materials are dry, that does create dust. Are those
Right.
also aerosolizing viruses if there was contamination?
It could very well do that. I mean, if you're creating any kind of an aerosol, if it's dry, again, we need some data on how long it can stay in what level of humidity environment and retain its infectivity, at least like a tissue culture infectivity.
I was talking to medical people and they were saying no misting. And they were talking about that for people in homes.
Oh, you mean like
the same type of misting, but it would be
A humidifier.
Like a humidifier?
Humidifiers
okay.
would be problematic. So from that, I thought, okay, that might not be good. But
I mean, I think that's for keeping them in the compost situation. I guess those misters are to keep the dust particles down. Yeah, but
and
not for this. Not for this application.
Right.
Jane, for a question for you about what happened to all in the compost of all the chicken mortality. What happened to all the compost?
It was all used for field crops, all used on the farms that it originated on. So these farms are in the middle of nowhere, a lot of them, and it all went to growing corn or whatever crop they were growing to feed.
Nice. A lot of questions about the PPE that you were talking about. Jen, do you have a list or kind of a more that we could have, or a descriptive list of specific PPE for specific situations?
We could put something together. I mean, just
I think. All right, let's work on that.
It wouldn't be gospel, but there is a video that I posted on there. That's when we were doing the fowl cholera. We tried to do the extensive PPE as best as we could so that people would understand the clean and the dirty and how you take off a glove, how you put on gloves, how do you undress. All of that is a big deal. And we have a couple of videos that do describe how to don and doff PPE.
Great. Note from Nellie Brown, who is with the Cornell Industrial Labor Relations. She's a safety specialist. She just makes a comment that they should not be safety glasses. It's goggles, unvented goggles that really protect the eyes.
Thanks, Nellie.
And so then Brenda points out, so given the dearth of PPE and the desperate the calls for PPE for medical professionals, does it make sense for community composters or for small scale composters that don't have the access to PPE to not be composting?
All my PPE was taken, all my masks were taken out of my storage room at Cornell. They did go to a hospital, it's okay.
But I guess that's a question we'll be on. So remember, we're coming on towards the end of this webinar. We do have three more webinars coming up, and especially the one in 2 weeks from now, we'll be specifically talking about small scale food waste collectors. And so I think we'll work so that in 2 weeks time, we can have a more definitive list of what kinds of PE would be recommended, and also not just PE, but procedures, because PPE is really, is important, but first you want to think about administrative protocols, technical changes you can make to limit exposure, and also want to just remind everyone that the primary route of exposure is person-to-person contact, sneezing, coughing. I mean, so while it is theoretically possible for a virus to contaminate food waste and theoretically possible for a worker to collect it, that's not the major threat that we're facing.
Right. Very true.
So just a reminder. Okay, we do have a time for a few more questions. I'm gonna close down at a quarter after the hour, so another 10 minutes. Steve and Carol asks, would reusable coveralls and reusable masks, like N95 neoprene with carbon filters, be good alternatives if you're not able to find the disposable ones? And if you provide a laundry room and showers so that you're leaving that stuff at work.
The laundry room and showers is almost critical. I'm going to jump back to farms again. To ensure biosecurity, there are a lot of farms that they buy the coveralls for people and they are laundered. They're taken off and laundered right on the farm. They get dressed on the farm, they shower. I know some compost facilities that do that as well, but most I'm not aware of. But I think it's really important. The virus can persist on your clothing. And I was reading in Johns Hopkins information that was shared with us, and they were saying, do not shake out your clothes, because then the dust comes off your clothes and then you're getting it. So get it right in the washer and wash with warm water and a good soap.
And that's a great recommendation, although just to warn you that Johns Hopkins recommendations that have been circulated widely over the internet are misattributed. Those are not, Johns Hopkins has disavowed that because there's some errors in that list.
There are some errors, yeah.
Yeah, so if you check Snopes.com, you'll see the details on that. But because I got forwarded, I was going to use it, and then when I checked it, because there were some errors, they said this is a misattributed set of recommendations. So be careful. Let's see what we got.
I saw one earlier when there was a discussion about vermicomposting, and is that okay for food? Under these circumstances, I would say the use of a thermophilic process first would be preferred because you'd have a definitive inactivation step if you get to the appropriate temperatures and time, and then you can follow that with the vermicompost.
Right.
I get, yes, and remember, we're talking about places that are accepting waste from other sites.
Yeah, I don't know
not
whether
home
people,
composting.
but I guess there are some places that,
Yes, there are commercial vermicomposters, and I think
Right.
you're right. That would be the appropriate caution.
Right.
We did post. We are going to post some recommendations for home composters where you're not taking stuff from outside the home, but everything is within the home, and that's a different situation, especially because it's typically months, if not many weeks,
it's a
if not many
long-term
months,
process. Yeah.
before you're going to harvest out. And so in that situation, you're
Yeah.
really just being careful as far as sanitation, hand washing, wear gloves, eye protection, all the basic protocols that you would take in a house.
And one of the things for home composting was make sure that you clean the handles of the buckets.
Yes. Yep.
You know,
Oh,
make
yeah.
sure you're thinking about that kind of stuff, because that's a surface again where you might have had it on your hands and it transferred to the bucket surface, and you know, then you're dumping the bucket out, so it could be problematic. So just think about those things, handles in general.
All kind of touch points.
I want to answer a question from Karen Dawson about sales. Actually, I'm not going to answer the question. I'm going to let her know that we'll be having our fourth webinar in the series will be on sales, on selling compost during this time, and how do you supply compost to consumers who are sheltering in place. How do you set up a sales area that's safe and keeps everyone safe, both your workers and customers? So that's something that we'll be covering at a later webinar. And with that, we are just about out of time, everyone. I appreciate.
Fred Michelle had a comment here. Why don't we read that one, Gary or Carrie, or I can.
Go ahead. I haven't seen it.
Viral RNA is very unstable outside of the protein coat of the virus. So naked RNA could not reassemble into an infective virus once outside the virus particle or an infected cell.
Right.
That's on everybody's chat.
That's fine.
Yep.
Yeah.
Okay, let me just share one last time here. And I just wanted to, again, let people know that we've got these slides coming, webinars coming up. Also, if you are interested in getting credits, if you're taking this, watching this webinar and are certified, in the email you receive tomorrow, you'll have instructions on how to get certification credits for today. And with that, I'll just say thank you for everyone to coming, and we look forward to seeing you again in future webinars.
Thanks, Carrie.
Thanks to my presenters.